Evolution is often referred to as a very slow process occurring over many millions of years. For this reason, evolutionary scientists openly acknowledge that large-scale changes, known as macroevolution, cannot be directly observed. Naturally, this leads to the question, if it is not observable, then on what grounds can anyone claim evolution is a scientific fact? The typical response is to claim that although we have never seen macroevolution, we can see evolution happening today on a small scale, known as microevolution. Microevolution involves tiny genetic modifications that result in superficial changes in an organism, such as an increase in a bird's beak size. These type of minor changes help the organism adapt to a slightly different environment. No one denies that this type of adaptive fine-tuning occurs through mutations and natural selection. However, proponents of evolution believe that given enough time, the small observable changes seen in microevolution will inevitably add up to large-scale changes known as macroevolution. This is the micro equals macro fallacy, which assumes macroevolution is essentially microevolution extrapolated over long periods of time without limit. This is an example of unbounded extrapolation, which is known to be extremely poor science.
Although in theory both types of changes involve the same underlying genetic processes like recombination, mutation, selection, genetic drift, and gene flow, there's a profound and essential difference between macro and microevolution that is often overlooked. Macroevolution requires vast amounts of additional genetic information, meaning the formation of many new functional DNA sequences, while microevolution does not.
According to evolutionary theory, all living creatures evolved from a single ancestral life form that lived billions of years ago, a microorganism believed to be similar to that of modern bacteria. As the first living organism to emerge from the primordial sea, it would have had minimal genetic information, just enough to carry out the essential life functions. A reasonable estimate for the amount of DNA contained in a hypothetical primitive bacterium would be less than 500,000 genetic letters, equivalent to about 2 500-page books of typewritten information. This may sound impressive, but not compared to humans with 3.2 billion DNA letters in a single cell, the equivalent of approximately 1,500-page books. Thus, in order for an ancestral microbial species to evolve into man, a mind-boggling amount of new functional DNA sequences would have to build up over time. This new genetic information would be essential in order to specify new functions and structures such as eyes, ears, brain, heart, lungs, and a multitude of other biological features found in higher life forms. The genetic specifications for even a single type of higher life form represent a large library of information. Yet there are millions of life forms: plants, sea creatures, birds, reptiles, and mammals, each with their own library of genetic instructions.
Evolutionists claim these new genetic specifications can arise through random mutations and the reproductive filter of natural selection. Textbooks offer a number of examples that are claimed to be direct evidence of evolution in action. Let's look carefully at a couple popular examples to see whether they involve the addition of new functional DNA sequences in order for macroevolution to be possible.
Consider the observable changes occurring within canines. Wolves, coyotes, jackals, dingoes, and the numerous varieties of domesticated dog are all believed to have descended from an ancestral wolf species. In textbooks, the development of the various dog breeds is cited as a demonstrable example of evolution on a small scale. McGraw-Hill's widely used biology textbook states this type of change will inevitably lead to large-scale evolution over time. In making this claim, the textbook authors are committing the micro equals macro fallacy, the blunder of employing unbounded extrapolation. The superficial differences seen in the various dog breeds are not caused by the formation of new functional DNA sequences that are required for macroevolution. Instead, the differences are caused by degenerative mutations in a small number of already existing genes. Mutations in these genes are responsible for the variation in traits like coat color, face shape, and foreshortened limbs. But such mutations are limited in the extent of change that they can produce before they disrupt the necessary function of those genes. Factor in the well-understood process of genetic recombination, the reshuffling of pre-existing genetic information, and the formation of the many dog breeds is easily explained. Although mutations have provided an additional source of variation for breeders to select from, this is not the type of change that macroevolution requires, since it has not resulted in any new genes or new genetic information. In fact, producing dog breeds through artificial selection results in an overall loss of genetic information. The non-desirable traits that are not selected are literally bred out of the gene pool. This explains why it is possible to breed wolves and select for certain traits over a number of generations to produce chihuahuas. But you can never breed chihuahuas to get back to wolves. The DNA information necessary to get back to a wolf has been lost.
So everyone knows that within the dog kind, we have lots of different breeds of dogs. And some people might say, well, if we can see all these different breeds of dogs, you know, long ears, long fur, short fur, certainly that's an example of new genetic information that's being added to have those traits. But when we observe this at the genetic level, that is not what we see. We're just seeing variation on traits that those organisms already had. The genes that code for fur in a dog, okay, they'll produce fur, long, short, different colors, things like that, but they're never going to become wings, okay? They're never going to become feathers. They're not going to change like that because they don't have that information to be able to do that, and there's simply no genetic mechanism that allows you to add the information that you need for evolution to occur. All you see is variation within the traits that they already have.
The modern theory of evolution is founded on the concept that beneficial mutations are the raw material for eventually building new sets of genetic instructions. One of the most popularly cited examples of beneficial mutation is the development of bacterial resistance to antibiotics. Standard biology textbooks claim it is direct evidence for evolution. But is true evolution really taking place here?
Certain classes of antibiotics are considered prodrugs, which means they do not pose a threat to the bacteria until they are activated into their reactive or lethal form through the assistance of enzymes naturally produced by the bacteria. However, random mutations in the bacterium have been shown to disable the function of the required enzyme, preventing the antibiotic from converting into its lethal form, resulting in resistant bacteria. Although these mutations may be considered beneficial, since the bacteria are now able to survive the antibiotic treatments, the genetic information that encodes an otherwise useful enzyme was lost. This is the opposite direction required for macroevolution. In the long run, these types of loss-of-function mutations are counterproductive to macroevolution. For this reason, bacterial resistance to antibiotics cannot be considered direct evidence for evolution.
One of the ways in which bacteria become resistant to antibiotics is through something called horizontal gene transfer. And this is something that is pretty unique to bacteria. Humans don't do it, animals don't do it. But what happens is that bacteria can essentially swap genetic information. So genetic information can be transferred, like an antibiotic-resistant gene, from one organism to another. But simply transferring genetic information is not what evolution needs. Just transferring it from one organism to another organism, and they're both bacteria, doesn't essentially change the essence of that bacteria. So that's still not the type of genetic change that we need to go from one organism to another. We have to acquire lots of new information, lots of new functional genes to go from one kind of organism to another kind.
For evolution to create higher life forms from lower life forms, there must be a very large increase in total genetic information. Additional genetic information is needed to encode the complex biological capabilities that characterize higher life forms. Any hypothetical large-scale evolutionary change would require vast amounts of new functional DNA sequences, which would have to arise through beneficial mutations filtered by natural selection. Although adaptive changes can occur through rare beneficial mutations, it is consistently at the expense of destroying DNA information, as seen with bacterial resistance to antibiotics.
There are many mutations that are advantageous. In that sense, they're beneficial in that they help adaptation to a certain circumstance. But when they actually look at what happened, the mutation almost always involves loss of function. Something's broken, a protein isn't functioning properly. So there's a disease called sickle cell anemia. The red blood cells of people with sickle cell anemia are misshapen. Instead of being a lozenge-like cell, they're shaped like a crescent moon or a sickle. And those deformed red blood cells can't carry as much oxygen as they should, and they're prone to causing blood clotting. Well, how is that beneficial? If you have this terrible disease, you're not as susceptible to the malarial parasite. So in certain areas where malaria causes lots and lots of deaths, you're basically trading one pathology for another. And so better to have sickle cell anemia, where you're sick but not dead, than to get malaria and die. So it's a very bizarre idea that sickle cell anemia is a beneficial trait. They use it because there's so few examples, convincing examples of beneficial mutations.
The information-eroding nature of beneficial mutations makes sense when we think about what mutations really are. They are literally random copying errors in the DNA. Think of them as genetic typographical errors. By their very nature, they disrupt cellular processes, such as gene regulation and enzyme function, by scrambling the organism's genetic instruction manual. They do not create information, they erode information bit by bit. This type of genetic change occurring over deep time will never result in genome-building evolution. It is going in the wrong direction informationally. Thus, the common argument that given enough time, a series of small changes, microevolution, will eventually add up to large-scale changes, macroevolution, is a fallacy. The rarity of beneficial mutations complicates things further. It is widely acknowledged in the genetics community that harmful mutations vastly outnumber beneficial mutations. These destructive mutations are eroding the information content of the genome at a much faster rate than rare beneficial mutations could build it.
So if you have bad mutations, even if just a fraction of them are accumulating, if the deleterious mutations are accumulating even at a very slow rate, and you have very few beneficials to counteract that, you're going downhill. The fact that beneficial mutations are very rare, and that most mutations are, if they're not neutral, they're deleterious, is one of the best attested facts of biology.